So, welcome back. In today's class, we will be learning about the double membrane organelle of a eukaryotic cell. In double membrane organelle of a eukaryotic cell, we have nucleus, we have mitochondria, and we have chloroplast. These are the three important organelles that we will be learning under double membraned organelles of a eukaryotic cell. To know about the characteristics of a eukaryotic cell and single membrane eukaryotic cell, please check out the another link that I have uploaded explaining these details. This video is continuation of that. In double membraned organelle, we have nucleus. We are going to learn about the structure and function of these three organelles. The most important part of a cell is nucleus. In nucleus, it is believed to contain the genetic material. The function of your nucleus is the storage of the genetic material. In eukaryotes, it is definitely DNA. It stores DNA and it provides it a suitable protection without denaturation. It also regulates the entry and exit of the molecules inside the nuclear envelope. To participate and carry out DNA replication, nucleus plays a vital role to regulate and carry out transcription. Here, nucleus is divided as various regions like your nuclear membrane. Here, this complete structure is a nucleus. Here we have the nuclear membrane, which is the nuclear envelope. This provides a protection for the genetic material. Then comes your nucleoplasm, which is believed to contain the chromatin in it, that is the genetic material in it. Then comes your chromatin, that is your genetic material, and your nucleolus, which is a hyperpigmented region of your nucleus. Then is your endoplasmic reticulum. As we discussed, it is attached to the nucleus, and since it has ribosomes in it, it is believed to remain beaded. That is why it is called as rough endoplasmic reticulum. When we take the genetic material, this is how a typical chromosome would look like. When we try to unwound them, it will look like this. The DNA is wound tightly together is called as chromatin. And when we unwind that, this is the linear structure of your DNA. It is wound around a protein called as histone protein. This will enable us to pack the large or very long genetic material inside a small cell. That is why, just like the thread has been wound around the wood, we have the genetic material being wound around the histone protein that will enable it to be compactly arranged within the cell. To see the structure or the function of the nucleolus, which is the hyperpigmented region of your nucleus, some chromosomes have sections of DNA to encode ribosomal RNA. A darkly stained region is the nucleolus. The ultimate function of your nucleolus is to make ribosomes. These DNA that will encode for ribosomal RNA is present inside your nucleus. The ultimate agenda of your nucleolus is to synthesize proteins, synthesize ribosomes, and ultimately ribosome synthesizes proteins. Ultimately, the secretory metabolites or the metabolic byproduct or a combination of amino acids, nothing but the protein. So protein synthesis is very important in a cell. So for which numerous ribosomes need to be produced, for which nucleolus is highly important because it synthesizes ribosomes. Ribosomes are the protein factories of the cell that synthesize large amount of proteins, and it is responsible for protein synthesis. Then comes the mitochondria. As we all have learned from the previous classes, mitochondria is observed as a powerhouse. This is believed to be true. This is a very important organelle of a cell because it is a evolved cell. It has to undergo a lot of complex metabolic activity simultaneously, for which more ATP is required. ATP is a ultimate energy currency that is produced by the cell. As in the by end of glycolysis, numerous ATP are produced. So for which for it to produce enough energy, mitochondria plays a vital role. Organelle, it has excuse me, it has been used to produce energy by the process of electron transport chain. Electron transport chain occurs in mitochondria. It also shows oxidative metabolism that helps us convert food into energy, to extract energy from food and thereby producing numerous amount of ATP. It is with the without the utilization of oxygen, since it is anaerobic method, or the mitochondria can afford to be bigger than cells without mitochondria. Without it, the cell cannot provide food, or will be able to get amount of in a required amount of ATP as per the requirement. The outer layer of your mitochondria is freely permeable, where to small molecules which contains a particular amount of concentration can enter inside the cell. But whereas the inner membrane is highly permeable in nature, which allows very very small molecules to cross the cell material. The matrix contains DNA. It also contains various enzymes, and where the TCA cycle or the Krebs cycle or tricarboxylic acid cycle occurs there. This helps in the conversion of the food into ATP molecules. Then comes the chloroplast. These chloroplasts are nothing but the oval shaped double membrane organelle. Here, outer membrane is believed to be the external surface, the outermost surface, and the inner membrane is just below that, within a thin intermediate membrane between the outer and the inner membrane, that is called as the stroma. Stroma is the space within the inner membrane. In vesicular plants, green algae, these thylakoids, I know there is a stacking up of thylakoids that are present on the topmost layer. Thylakoids involve chlorophyll and carotenoids. The chlorophyll is the important pigment that chloroplast contains. Chloroplast is like a sac or a apparatus inside which chlorophyll pigment is present. Chloroplast is an apparatus. Chlorophyll is the pigment. That is why we call it as a pigment apparatus, or pigment containing apparatus. This is called as a pigment containing apparatus, where chloroplast contains chlorophyll pigment within it. So this is how an ultra structure of a typical chloroplast looks like. These chloroplasts are present in photosynthetic organisms. Photosynthetic eukaryotic organisms contain chloroplast. Here, apart from chlorophyll, they also contain certain accessory pigments like carotenoids, beta-xanthin. All these are accessory pigments where chlorophyll is the main pigment. As we discussed, it has the outermost membrane and the inner membrane. Inside the inner membrane is the stroma, and it has a pile of coins, stacking up of the coins, or pile of coins arranged. That is nothing but your thylakoids. Each coin or the stacking up of the structure is called as thylakoid. Together, it is called as grana or granum. That is the stack of thylakoids. Inside your thylakoid is your chlorophyll pigment that is present. And the last one that we are going to see is peroxisomes. Yes, peroxisomes are actually in single membrane organelle. Peroxisome is a single membrane bound organelle, which is small round organelle enclosed by single membrane. Peroxisome are single membrane bound organelle. Here we have to understand that to nullify the effect of the toxic byproducts produced by the product of the metabolism, or toxic molecules entering inside the cell, or when intermediate byproducts are toxic, that has to be nullified by the